Good evening, students. This is Prafull. Welcome to One Page Biology. So today we are going to discuss one of the question papers of NEET Biology. This question paper is of 2023 year, and today I'm going to discuss some of the questions of section A, that is botany part of the NEET Biology question paper. So let us see the questions one by one. We can attempt all of them, and I will try to solve few of them in today's video. But of course, in my coming videos, I will keep on discussing some important MCQs with you all. So let's begin with the discussion. Now, as we can see that, dear students, here section number A, always botany part. So here the first question is straight away, as you can see here. I hope it is clear, visible for you all. The question is, which of the following is not observed during apoplastic pathway? So now, dear students, this particular question, if you recollect, this is from the chapter Plant Water Transport. So this is from 11th standard NCRT biology textbook. That is from the paper 1, that is botany section. So this is a good question, which is that which is not observed during apoplastic pathways. Now, dear students, if you recollect that in plants, there is of course transport of water and minerals and transport of food as well. But specifically when we talk about transport of water, there are two basic pathways through which the water actually is transported in plants. One is called as apoplast pathway, and the other is symplast pathway. So both pathways help in the transport of water. But what is the difference between the two, that is apoplast and symplast? So we all know that in apoplast pathway, the water actually travels from the extracellular spaces, that is from the cell walls, and this is mainly because of the phenomenon called as imbibition. What is imbibition? Imbibition is attraction of the liquid molecules towards the solid molecules. Especially here, when we talk about the solid molecules, we are talking about the cellulose, which is present in the cell wall of the plant cells. And cellulose is of course hydrophilic in nature. Hydrophilic means what, dear students? It means that it attracts water. So one of the pathways is apoplast, in which the water is actually traveling from the cell walls. It is not entering from inside the cells, but it is actually going from cell walls. So this is one of the pathways. So this majorly happens in the root section. That is, if you talk about the roots, then we when we study the anatomy of roots, we know that there are root hairs, the outermost layer, that is epidermis. We have root hairs and dicot stems, of course. So root hairs are there, and then we have different layers which are present in the inside the root. So we have basically the epidermis, the cortex, and then we have epidermis. Then we have endodermis. Then comes the pericycle, and the pericycle, after that we have vascular bundles, that is phloem, xylem, and finally then there is pith. So when the water is actually traveling from the inner parts of the roots, that is mainly from the epidermis till the majorly the endodermis. So this majorly happens through the apoplast pathway. And once the water reaches to the endodermis especially, which is basically the layer of cells. These are barrel shaped cells. Barrel shape means these are cylindrical cells. So here the water gets blocked. Why it gets blocked? Because in endodermis the cell walls are made up of a particular material, that is a wax-like substance called as suberin, and here the water get blocked. And from here further, that is from endodermis inside the roots, that is why the pericycle or the vascular bundles, the basically the phloem and the xylem, and then it reaches to the pith. But inside the root, that is from endodermis to the pith, it occurs via the symplast. And symplast is majorly the way by which the water actually passes through the inside of the cells through intercytoplasmic connections. And what are those intercytoplasmic connections? These are known as plasmodesmata. So coming back to the question, dear students, the question is saying that which is not observed in apoplast. Now see, very simple. We have to in these questions you have to be quite careful, dear students, because the question is saying not observed. So something which is not observed in apoplast, right? So let's read the options. The option says that first is apoplast is continuous and does not provide any barrier to water movement. Yes, of course it is true, because water is actually traveling from the cell walls, and as we said, it is going from cell walls, so there is no barrier to the movement, and it is a continuous movement. So option number 1 is absolutely true. So this is not the right answer, right? Because we have to go with the wrong answer. Second option is movement of water occurs through the intercellular spaces and walls of the cell. Just now we said yes, water movement is from the cell walls. So second is also right. Third option, movement does not involve crossing of cell membrane. Movement does not involve crossing of cell membrane. This is also true because, of course, since the water is going from outside the cells, since it is traveling from the cell walls, there is no question of cell membrane. So, of course, the water is not crossing the cell membrane, right? So, option number 3 is also correct. Fourth option, the movement is aided by cytoplasmic streaming. Now, dear students, here they are saying that the movement is aided by cytoplasmic streaming. Where is cytoplasm present? Cytoplasm is present inside the cell. So, is hypoplast pathway occurring from inside the cell? Not at all. Just now we said that the water is going from the cell walls. So option number 4 is absolutely wrong. It is not observed in hypoplast pathway. So hence the right answer to this question will be the fourth option, that is movement is aided by cytoplasm. This is not observed in hypoplast pathway because this will be observed in symplast pathway, dear students, because there the water goes from cell to cell via the cytoplasm. Okay, I hope this is clear to you all. Let's go and read the next question. The device which can remove particulate matter present in the exhaust from the thermal power plant is option number 1 catalytic converter, option number 2 STP, option number 3 is incinerator, option number 4 is electrostatic precipitator. So dear students, this is a straightforward question. So I think here there is no concept involved. They are just asking which is the device which helps in removing particulate matter from a thermal power plant. So there are various ways by which the waste which is produced in the factories. So the question number 103, which of the following never occurs during mitotic cell division? Very important, dear students. Cell cycle, that is the chapter number. So very important chapter, cell cycle and cell division. Again from 11th standard NCERT. The question is, which one of the following never occurs during mitotic cell division? Never, again. So dear students, always you have to be careful about such questions because there is a high chance of some error happening, because sometimes we tend to be a little hasty while attempting the questions. So always make sure that you read the question very very clearly, and ensuring that you read each and every word properly. So here they are saying that which is not observed in mitosis. Let's read the options. Okay, coiling and condensation of chromatids. Of course, yes, it is observed in mitosis because, first of all, in mitosis, the basic thing first is the interphase, which is basically called as the preparatory phase, wherein the cell prepares for cell division. And you all know that in interphase there are three subphases, that is G1, S, G2 phase. Then starts the mitotic phase, and in mitotic phase there are four majorly phases, which are basically nothing but the nuclear division, that is the karyokinesis, which involves prophase, metaphase, anaphase, and telophase, which is followed by the cytoplasmic division. So in the first phase, that is prophase, pro means first. In that phase itself, what is happening, what is getting what kind of changes are occurring inside the nucleus of the cell? Very first thing happens is the nuclear membrane disappears, or you can say it gets dissolved. Second is second is, of course, the chromatin fibers which are present in the nucleus, dear students. They start condensing. They start becoming condensing because of dehydration, and dehydration is basically loss of water. So as they start losing out lot of water, basically they will start becoming thicker, and as they become thicker, they become shorter and more of condense, and they take the shape of chromatids. So basically you can see in the prophase they have started becoming more and more thicker, and they are started looking like somewhat like chromosomes, but not exactly chromosomes, because proper chromosomes we can very clearly see in the next phase, that is in the metaphase, wherein they properly become thick, coiled, condensed. At the same time they come near the the center of the cell. But in prophase they have just started becoming condensed, and that's why, of course, the first statements, that is coiling and condensation of chromatids, that will be right for mitosis. But as we have to say that, yeah, the question is asking us never occurs, so of course first option is not right. Second option, let us read. It says that spindle fibers attach to kinetochores of chromosomes. What are kinetochores? Kinetochores are basically certain proteins which are present in the centromeres of chromosomes. So whenever the cell is dividing, especially here we are talking about the majorly the plant cells, dear students, because animal cells lack centrioles. As you all know that animal cells are without centrioles, and there the formation of spindle fibers, they occur directly from the cell membrane. So, but in case of plant cells, since there are centrioles which are present, so centrioles also divide during the S phase, and they also move towards the opposite poles of the cell. And what is happening from the centrioles? From centrioles, there are special kind of fibers which are formed. These are spindle fibers, and these spindle fibers attach to the chromosomes. And where do they attach? They attach to the centromeres. And on the centromeres, there are special kind of C-shaped structures which are made up of proteins. These are known as kinetochores. So the spindle fibers attach to the kinetochores. With the help of spindle fibers, all the chromosomes arrange at the center of the cell, that is, which is considered as the equator of the cell, equatorial plate of the cell. It is also called as metaphasic plate. So dear students, second option is right. Third option, movement of centrioles towards opposite poles. Just now we said, yeah, yes, it occurs. As far as the plants are concerned, centrioles are present and they will move towards the opposite poles. Animals, no centrioles, right? But let us see. Let's assume that the question, sometimes in such questions you have to assume what the examiner is wanting to say. It can't be, very much straightforward, right? So let's see what is the fourth option, and then we will decide which is the most correct option. Always remember, you have to go with the most correct answer. Last option is pairing of homologous chromosomes. They are talking about homologous chromosomes. Pairing of homologous chromosomes. First of all, what do we mean by homologous chromosomes? You have to consider basically a cell which is diploid. Of course, mitosis is occurring where? It's occurring in every cell of our body. So whether it is a plant cell or an animal cell, the cell is diploid, right? So in mitosis, what happens? In the prophase, we said the condensation of chromosomes is taking place. Then in the metaphase, all the chromosomes are coming at the center, right? In the anaphase, the sister chromatids are going to be removed, right? They are going to move away from each other, and ultimately in the telophase, all the sister chromatids move towards the opposite poles. Again, there is formation of nuclear membrane. Again, the basically the hydration of chromatids will occur and they will convert into chromatin fibers, and as a result of which there will be formation of two new nuclei in the cell. Option number 4 is saying pairing of homologous chromosomes. Is this observed in mitosis, dear students? Think about it. The answer is no, because in mitosis there is no pairing of chromosomes. Where does pairing of chromosomes occur? That happens in meiosis, because in meiosis, if you recollect, dear students, meiosis is basically divided into two substages, that is meiosis 1 and meiosis 2, right? And majorly in meiosis 1, especially during prophase 1, which is the longest phase of meiosis. So prophase 1 is further divided into leptotene, zygotene, pachytene, diplotene, and dikinesis. So in case of basically the leptotene, zygotene, that is zygotene, zygous means pairs. So dear students, in zygotene especially, you will observe that the homologous chromosomes are getting paired. What do you mean by homologous? That is the maternal and the paternal. That is, as we all know, that in our body, in total, the total number of cells in every cell of our body is, total number of chromosomes in every cell of our body is 46. That means 23 are maternal, 23 are paternal. So these chromosomes, which are nothing but the maternal and the paternal, maternal means which are coming from mother and paternal means which are coming from father, they pair actually in the zygotine phase of meiosis 1. And that's how the 23 pairs are formed in the zygotine phase. So this is observed in meiosis, dear students. And that's why that is the speciality of meiosis, that is the uniqueness of meiosis, because in meiosis you can also see that there is, in the third phase of basically the prophase 1, that is pachytene. What is observed in pachytene? There is crossing over, and there is crossing over, dear students, between the non-sister chromatids. Getting my point? So whenever these homologous chromosomes pair, that is the maternal and the paternal chromosomes pair, the non-sister chromatids undergo overlapping, or there is crossing over between them, and that point where they cross over, that complex is also called as synaptonymal complex. So that is very, very important, because that is mainly responsible for variation, because that event leads to the exchange of the chromosomal genes, or you can say the chromosomes, and that is mainly responsible for variation. So option number 4 here is incorrect, because pairing of homologous chromosomes is not right as far as mitosis is concerned, and that is not taking place in mitosis. So the right answer will be pairing of homologous chromosomes. So dear students, observe, this was a good question, because many students would have felt that option number 3 is right, because if you consider animal cells, there are no centrioles present in them. But as I said, always read all the options. Don't ever try to be in hurry. Make sure you read all the options and go always with the most correct answer. Let's read the next question. The next question is, this is again a straightforward question, dear students. Again, hydrocolloid carrageen is obtained from hydrocolloid. Hydrocolloid means it's a gel-like material, and it is hydrophilic in nature. Carrageen is a type of a polysaccharide. So where it is present? They are asking us options are Phaeophyceae, Chlorophyceae, and Phaeophyceae. Option number 3, Phaeophyceae and Rhodophyceae. Option number 4 is Rhodophyceae. So these are all three types of algae. We all know Chlorophyceae is green algae, Rhodophyceae is red algae, and Phaeophyceae is brown algae. So here the option is directly straightforward. The answer is Rhodophyceae, because Rhodophyceae is red algae, and in red algae basically there is presence of carrageen. Okay, so option number 4 is right. Okay, let's go with the next question, dear students. Question number 105. Read the following statements about the vascular bundles. Okay, so you have to go with the correct statements. You have to see that how many of them are right out of how many statements are given. 5 statements. Okay, so out of 5, which ones are right? You have to identify them. Let us see. First statement: in root, xylem and phloem in vascular bundle are arranged in alternate manner along the different radii. Yes, it is true, because we all know that in roots, basically the arrangement of vascular bundles is radial. What do you mean by radial? That is, the xylem and phloem are present on the different radii in an alternate manner. Such arrangement is called as radial. Second option. So first option, as we said, it is right. Second option: conjoined closed vascular bundles do not possess cambium. Conjoined and closed. See, again, important. What do you mean by conjoined? First of all, as we just now said, that there are two types of arrangement. The first option was talking about radial arrangement. So that we said vascular bundles are present on different radii. Second option, that is, now here they are talking about the another type of arrangement, that is conjoined. What do you understand by conjoined arrangement? Dear students, in conjoined arrangement, vascular bundles are present always as a bundle. That means that they are present together as a single bundle. That means the phloem and the xylem are present as a single bundle, and depending on whether cambium tissue is present between them or not, they are of two types. One is closed conjoined bundle, another is open conjoined bundle. If it is closed conjoined bundle, then cambium is absent. So if it is open conjoined, then it is cambium is present. Closed conjoined, cambium is absent. So here what they are saying is conjoined closed vascular bundles do not possess cambium, right? Because they are saying about the closed conjoined bundles, right? Option number 3. In open vascular bundles, cambium is present between. See the same type of similar type of statement. Now here they are talking about the open. So in open, as we said, in open conjoint, cambium is present. Okay, fine. Option number D. The vascular bundles of dicot stem possess enlarged protoxylem. Okay, vascular bundles of dicot stem possess enlarged protoxylem. Dear students, again, fourth statement is absolutely right, because we all know that, see, in stem, we always see endarch type of arrangement of the xylem. What do you mean by endarch? Endarch means the protoxylem is present inside, metaxylem, which is formed later, is present outside. Whereas in case of roots, which type of arrangement is there? It is exarch. In exarch, we see that protoxylem is outside and metaxylem is inside. So option number 4 is also right. So A was right, B was right, C was right, D was right. My God. Now let's check whether at least one has to be wrong, right? So generally one is always right, wrong. So the fifth option is, in monocots root usually there are more than 6 xylem bundles present. So the fifth option, that is E, in monocots root usually there are more than 6 xylem bundles present. This is also true. So one of the options. So, oh, now here, see, this is, I think, an ambiguous questions. Dear students, such questions, there are chances always of certain questions being ambiguous in nature in a NEET exam. So I believe that here all 5 options are correct, but if you ask me, then I would say that A is absolutely right, B is absolutely right, C, they are saying that in open vascular, okay, the more right way of saying this would have been in open conjoint, but still if we consider C as right, then A, B, D and E, but there is nothing, there is no option like that. Okay, so let's consider this as a ambiguous question, dear students, because I think here A is right, B is right, D is absolutely right, and E is also right, because they are saying that monocot there are usually more than 6 bundles. This is basically a polyarch condition. We all know this is a polyarch condition. Polyarch. So this has to be right, right? There is no question about it. So this is a bit of a ambiguous question. I consider this as an ambiguous question. Let's see. Okay, anyways, this is an ambiguous question. We'll check it later. Okay, let's move on to the next question. See, whenever, okay, fine. Since it's good that we encounter such question, at an early stage. See, these things do happen in NEET question paper also. Whenever we will be actually giving you a NEET exam, go ahead, because there are 90 questions, dear students, and you have to get all. As far as biology is concerned, you have to ensure that you get all the questions as the right, answers. ठीक है, चलो नेक्स्ट क्वेश्चन पे चलते हैं. The question is saying that DNA polymorphism forms. See, straightforward. What is DNA polymorphism? Change in the DNA. And whenever we talk about the changes in the DNA, dear students, here we are talking about the polymorphisms. What do you mean by polymorphisms? Dear students, these are certain sequences in the DNA which basically show the changes. When we talk about the changes, that means these are unique sequences in the DNA. So you have heard about the satellite DNAs, dear students. So satellite DNAs are the ones which are unique sequences. These are basically different. These are unique in every person, and they, depending on their size, depending upon their overall repeat sequence, there are different types like the mini satellites, the microsatellites. So one of the type of satellite DNAs is VNTRs. I hope you have heard about VNTRs, because VNTRs are been considered, they are been used in the DNA fingerprinting technique. What is the full form of VNTR? VNTR is variable number tandem repeats, and that's why, dear students, they are being used in the matching of DNA in the technique called as DNA fingerprinting. So the VNTRs are matched, and if the VNTRs are matched between the parents and the children, there is a high chance that that child, or the child is belonging to that particular parent, and this is often the technique used in paternity testing, right? Of course, in also the crime cases. So DNA fingerprinting is the right answer to this question without any doubt. Fourth option is saying that both genetic mapping and DNA fingerprinting, both genetic mapping, but genetic mapping, dear students, is a different thing altogether. Gene mapping is mainly done to find out the order of genes, the location of genes on the chromosomes. But that doesn't involve, that has nothing to do with the changes in the DNA. So the right answer for this is option number 3, DNA fingerprinting. Let's attempt one more question, dear students. So I'll try to keep it short video, because we will try and discuss every time around 5 to 6 questions, because that will make sure that the video is shorter, and you can also quickly go through that video whenever you feel it like going through. Okay, next question is, okay, this is like again good question based on assertion and reason types. So you all know that such questions, you have to identify whether the first statement is that assertion is true and reason is also true supporting the assertion. Sometimes, so both are true. Sometimes one is true and the other is not supporting the other statement, that is, as a reason is not supporting the assertion. Sometimes assertion is incorrect but the reason is true, that might also be right. And the fourth possibility is both are wrong. Okay, let us see which is the right option. Assertion is saying that polymerase chain reaction is used in DNA amplification. So good, that is a very simple statement, dear students. We all know PCR technique. PCR is a technique by which the DNA is amplified. the segment of the gene amplification method, dear students, you can get multiple copies of DNA, multiple copies of gene fragment with the help of this particular technique, right? So assertion is right. What is the second statement saying that reason is saying ampicillin resistant gene is used as a selectable marker to check the transformation. Ampicillin resistant gene, uh, of course, this is also one of the ways by which you can identify the resistant bacteria, and this is something which you do to select the transformants from the non-transformants. So you all know about the technique that is transferring the that is the plasmid, and inside the plasmid there is an ampicillin resistant gene. There is also tetracycline resistant gene, right? So of course all the bacterial cells, when you grow on a medium, on a suitable medium, so of course if you want to select the ones which have successfully transformed, that is the ones which have successfully taken up the plasmid. So how do you know them? So if they are resistant, if they are growing on ampicillin containing medium, so they are transformed, because if they have, the vector has the ampicillin resistant gene, and those which are not able to grow, they are non-transformants. But is the reason supporting the assertion here? No, because there is no relation between the ampicillin resistant gene or the transformation technique and the PCR. There is no connection between the two. So here, which option do you think will be right? The option which says that assertion is right, reason is also right, but it is not supporting the assertion. See, dear students, in such questions you have to be careful. So let's read the options first. Let me see which one. So let me read the option. The option number one is saying that A is not correct but R is correct. No, A is correct. Of course, A is correct. PCR is a gene amplification technique, or rather DNA amplification is used. Second option says that both A and R are correct, and R is correct explanation. Both A and R are correct. Yes, A is also correct, R is also correct. But is R the justification of A? Is R supporting A? Not at all, because there is no connection between transformation and DNA amplification. No, absolute no connection. So second option is also wrong. Third option, both A and R are correct, but R is not correct explanation of A. Absolutely right. This is what we are trying to find out. Dear students, this is the option which we were looking for, right? So they are saying that A is also right, R is also right, but R is not supporting A. R is not the right reason for A. So option number third is right as per our knowledge. Let's see whether the fourth what they are trying to say. See, the fourth is A is correct, R is not correct. Wrong. Both A and R are correct. So right option will be 3. Okay. So be careful about such questions. So dear students, how many questions did we solve now? 101, 2, 3, 4, 5, 6 and 7. So 7 questions we have solved. So I hope this video was helpful for you in your NEET preparation, dear students. If you have any doubts or suggestions, let me know in the comment section. And all the best for your NEET preparation, dear students. Keep solving maximum questions. Stick to NCERT textbook, and whatever doubts you have, let me know. So see you all in the next video. Take care of your health, dear students. Thank you so much. This is Prafull, and hope to see you all in the next video with some other biology related concept. Thank you so much. Bye.